Dear students, today's topic is anti-HIV drugs. Let us start with definition of anti-HIV drugs. Agents that are used to treat AIDS or stop the spread of HIV infection. Human immunodeficiency virus is the cause of acquired immunodeficiency syndrome. Both HIV-1 and HIV-2 cause AIDS, but HIV-1 is found worldwide. HIV is the human T-cell lymphotropic retrovirus which infects and kills helper T lymphocytes, resulting in loss of cell-mediated immunity. This is the microscopic image of HIV. HIV is called a retrovirus because it works in a back-to-front way. Unlike other viruses, retroviruses store their genetic information using RNA instead of DNA, meaning they need to make DNA when they enter a human cell in order to make new copies of themselves. HIV is a spherical virus. The outer shell of the virus is called the envelope, and this is covered in spikes of the glycoproteins 120 and glycoprotein 41. Which allow HIV to lock onto the CD4 receptor on CD4 T cells and enter the host cell. Let's discuss the life cycle of HIV. As you can see in picture, the life cycle of HIV is described in various stages. I am going to discuss each stage. At first, the stage the attachment and entry of HIV take place. The process of producing new viruses begins when HIV gains entry into a cell. This process happens in two stages, attachment and fusion. HIV infects immune system cells which have a CD4 receptor on the surface. These cells include T lymphocytes, also known as T cells, monocytes, macrophages, and dendritic cells. The CD4 receptor is used by the cell to signal to other parts of the immune system the presence of antigens. When HIV makes contact with the CD4 cell, the GP120 spikes on the surface of HIV lock onto the CD4 receptor and another co-receptor, either CCR5 or CXCR4. The GP41 protein is used to fuse the HIV envelope with the cell wall. This process of fusion allows the HIV capsid to enter the CD4 cell. Several types of antiretroviral drug have been developed to block different stages of the processes of attachment and entry. The GP41 and GP120 proteins on the surface of the virus are also targets for vaccines that are designed to produce antibody responses. The next stage is reverse transcription process. When HIV RNA enters the cell, it must be reverse transcribed into proviral DNA before it can be integrated into the DNA of the host cell. HIV uses its reverse transcriptase enzyme to convert RNA into proviral DNA inside the cell. Two types of antiretroviral drug have been developed to stop the action of reverse transcriptase and the creation of proviral DNA. The nucleoside and nucleotide reverse transcriptase inhibitors block HIV production by inserting a nucleoside or nucleotide into the chain of HIV DNA as it is created, terminating the chain. Next stage is integration. After HIV RNA is converted into DNA, the HIV integrase enzyme attaches itself to the end of the proviral DNA strands and it is passed through the wall of the cell nucleus. Once the proviral DNA enters the cell nucleus, it binds to the host DNA and then the HIV DNA strand is inserted into the host cell DNA. HIV integrase inhibitors have been developed to block the transfer of the HIV DNA strand into the host cell DNA. After the proviral DNA is integrated into the DNA of the host cell, HIV remains dormant within the cellular DNA. This stage is called latency, and the cell is described as latently infected. It can be difficult to detect these latently infected cells even when using the most sensitive tests. The next stage is transcription and translation. The cell will produce HIV RNA if it receives a signal to become active. CD4 cells become activated if they encounter an infectious agent. When the cell becomes active, HIV uses the host enzyme RNA polymerase to make messenger RNA. This messenger RNA provides the instructions for making new viral proteins in long chains. The long chains of HIV proteins are cut into smaller chains by the HIV protease enzyme. The next stage is assembly and budding. These protein chains begin to assemble into new viruses at the cell wall. As the virus buds from the cell wall, its genome becomes enclosed in a capsid produced from HIV's GAG protein. After the new virus is assembled, it must leave the cell by pushing through the cell wall. To leave the cell completely and become infectious, the virus must take lipids from the cell wall to make the surface glycoproteins. Maturation inhibitors are being developed to block the cutting of the GAG protein that is needed to produce a mature virus. The upcoming topics are: classification of anti-HIV drugs, synthesis, mechanism of action and use of anti-HIV drugs, brand name of drugs,